Okay. Right. The last time we discussed few things. That's gene expression control. Prokaryotes and eukaryotes, gene expression control. I think one of the interesting topic we also touched a little bit. That is if the genotypes, the genes are exactly same, and how this gene change, like a twin study. If we study in twins, I think that just can watch this one a little bit. This the video of this twin study. If we little bit notice when the twins are very young, they have very close, exactly the same, but then lifestyle is changing because they can marry, but they can divorce while still having the marriage, and the shape is changing. Maybe so much of differences, like they select different job, their behaviors are different, and also there are changes. What we look from outside is also different. But if you look very young stage, we cannot identify so much of differences behaviorally, and also the looks are same. But when the people are growing, twins are growing to the other stage. Can you see the screen, the videos? I think you can see, right? Am I?
Yes, sir.
Okay, right. Then scientists look genetic level, what is the difference? So they're trying to find it out. What is the difference between genes level? Okay, one thing maybe we can understand, if we see some kind of a different in twins. For example, if you look this picture, right? You might argue, okay, this may be due to how they want to change. If somebody wants to change their hairstyle, and somebody wants to make a tattoo, other doesn't want to make a tattoo, and the clothes and the beard, like they want to have a beard like different way. This may be the changes. So this may be nothing to do with genes, right? If you look this picture, like very carefully observe, you can see still changes
The
of
shape of the face is different.
shape. Yeah, exactly. The face has some changes. But still, if you argue that this may be due to because of the nutrition, because if they easily bit of fat, then get a round shape, maybe like that.
No,
we-
that's
yeah, okay. The different shape. Okay, you see, they have a not exactly like something externally doing it, but it's like a differences, right? We see the differences. Here is another picture, but if you look the small age, it's very hard to find the differences. But when they grow up, we can see the difference. And also, if you look this kind of a thing, like these are some of the example given. When they live in different places, so they were born as twins, but after that they separated. Maybe they live in different places, and it brings some kind of differences, right? Okay, coming back. coming back to address the question, if we have a still doubt, the doubt is their genes are not changed. Okay, we can't see that one. We see the phenotype is change. Change of the phenotype, right? The phenotype is the appearance, how we can see them. The what we can observe is the phenotype, and the phenotype is decided by the genotype. But when the twins they were born, they have the same genotype, right? Twins, they have the same genotype. Example, let's say this is a twin A, this is a twin A1. If you look chromosomes location of the gene, if they have a and simple A, that's capital A and simple A. There's no change. You don't see change of the genotypes, right? Capital A, simple A, capital A, simple A. And then you analyze all the genes, you can't find the differences. You don't see any differences. But then how we can explain when they grow bigger, how this phenotype is changing? If there's no change in the genes. Right, it's a bit difficult things to explain or maybe answer these changes, but when scientists study, they came up again the same conclusion. There is no change of the sequence of the DNA. DNA is, the sequence is not changed. DNA sequence is not changed. Okay, then there is one difference they identified. The difference is like this. Let's say if there is a gene and this gene expressed to make this protein. The gene expressed to make the protein. And the other, when it's again the story of twins, this gene is not expressed and there's no protein. Now I think this is a little bit understandable. This sequence is same. They have all the genes, the sequence is same, but we can understand this gene make the protein, this gene doesn't make the protein. So that is one explanation to address the issue, the differences of the phenotype. Then when we go into a little bit more about this situation, why the gene is not expressed. The gene is not expressed because of the influence from the environment. So then if they exposed at two different environment, then they have different conditions. For example, there was a small story one time. The renal kidney disease, because kidney disease is quite common chronic kidney disease in some part of the country. And there were twins. One twin has a problem with the kidney failure. The other one has no problem. So then easy the other twin can donate the kidney, right? So that's again explain environment exposure can change. Now you can easily understand if they exposed to some toxicities, the kidney can damage. Other one not exposed to the toxic compound, the kidney is not damaged. So I think easily understandable, some factors from the environment can change the phenotype, right? So this is come under the topic called epigenetics, right? So it's the story now, all the things place into the epigenetics. So today we're going to learn this epigenetics, right? Sanjuna. That means they fuse together.
Yeah, like both like Edexcel and together, like they're attached. And outside of that, but they got two hearts, but the same legs. I saw this documentary on YouTube where there are two girls in Australia.
Yeah, we see a situation like twins when they're born. There are some situations, mostly they fuse like a head region.
Yeah.
They're fused together, like they have some kind of a strong connection between each other. I think some of the situation doctors can make a surgery, and if it is a little bit less connection, they can remove them. This may be a yes, Sanjun. Okay, we're just talking about this new lesson, epigenetics. I think to find it out why they are joined together when they grow early stage, twins starting points of identical twins is zygote divided into two, and usually zygote grow as a one, but in this situation they divide into two and one complete to make a one organism. Other one zygote is also growing to make another zygote. So during this development stage, if they are very close, like it's not separate in the uterus, and they're very close and they start to grow very close with each other, I think there's a chance of fusion of the cells because they are same cells, right? So then this may be the explanation if they are not separated in the uterus. Let's say this cell is implant here, the other cell is implant here, then they grow as separately, but they are very close, like here and here. There's a chance of again combination of these cells. Right now we understood the phenotype is the observe character, which is decided by the genotype. But when the twins born, they have the same genotype. When they're exposed to the environment with the time, they have a different phenotype. This is explained in a broad way. They have a same genes, but this gene express to make a protein, and this gene does not express. It's not the sequence. It's all about the expression of genes, whether they expressed, frequently expressed, rarely expressed, not at all expressed, is the reason they can change their phenotype. And this is strongly influenced by the environmental factors. So this new kind of information we can discuss under epigenetics, right? So epigenetic is change let's work here. Okay, right. I write here then epigenetics, right? The definition of epigenetic is a change of the gene expression without change of the gene sequence. If we look, gene sequence does not change. It's exactly the same. For example, we know the gene sequence decided by the nucleotide, how they organize, like A, C, T, G, A. So this has no change. Let's say the identical twins. If we take the next one, it's also same. This should be T, A, C, T, G, A. So that's the first and the second one. You don't see any changes, right? The nucleotide ACT, GA, ACT, GA. But what will happen? This may be protein expressed here, and your protein is not expressed. So then, without the change of gene sequence, gene expression change. When we talk about the gene expression, yes, identical twins, they have same gene sequence, no change. Identical twins have same gene because it is coming from single cell. Zygote is single cell, and they divide exactly by mitosis to make two cells. That sequence does not change. But the expression change, right? Expression in the sense, last time we learned the gene expression control. So expression start, the promoter region, that's a gene. We learned the binding of transcription factors, then the binding of RNA polymerase, then the gene transcribed into a mRNA. Then the mRNA splice, exon and intron splice, and then only the exon join to make the mature RNA, messenger RNA. And the mature messenger RNA should go to the ribosome. Then they make the protein. Now you look all these process, the transcription factors, RNA polymerase binding, and then making messenger RNA, splicing, and then make mature mRNA, and the mature mRNA make the protein. It's a long process. Imagine if something influences this process that cannot make protein. That's why I'm telling you that this gene sequence is not changed. But it's not only the gene sequence, there are many events between. Transcription factors, the many transcription factors should bind, RNA polymerase should bind, all these things control the process. Now, we can understand if any influence from the outside change this process, cannot make the protein. I think epigenetics, the concept is the change of this gene expression at any point. Then protein may express, or protein not express at all, right? So if you look at your notes, we can see the definition, the study of change in organism, the study of change in organisms caused by modification of gene expression rather than alteration of DNA sequence itself. So page 58, you can see it, right? 58. The study of changes in organism caused by modification of gene expression rather than okay. Why don't you add this note, like this one? Yeah, that space is there. So gene expression you can show here. That's a promoter. I think I will show it like here. Promoter. This is the gene, and these are the transcription factors. This is the RNA polymerase, and this is the pre-mRNA. And then this is the mature mRNA after splicing, and then it goes to the cytoplasm to make the proteins, right? So in epigenetics, if we take this whole complete process from here to here, there is a change. Change in expression, right? Anywhere it could be change in this expression. Sequence, no change.
Like what if those like transfer to children, like they have the same gene for the back, like
No, if the twins, when they get the children, then their father is different. Like if mothers are twins, when they marry different fathers, father is different. So different genetic information coming from the father. But if we assume a situation, fathers are twins, mothers are twins. When they marry, but still children's DNA is different.
so like is there any possible way to find out whether that father's child is him because they got the same gene?
Well, I think I see somewhere in the internet, doctors say don't marry twins, like father twins, mother twins. They say don't marry always this combination. Probably it's difficult to find which one is a biological father in some cases, right? It's not that easy to find father, because uh, the twins, the son, when they have a children, their DNA is different, right? That means, let's say, this one is a one marriage, and this one is a one marriage, right? And they are twins, like these two men twins, woman twins. When they have children, it does not necessarily the same. When they may got a girl, but this family got a boy. But their DNA is different. This case, DNA is different.
Yeah, but the dad's DNA for the children is the same, right?
Yes, DNA is coming from mother, and DNA is coming from father, like half, right? And here DNA coming from mother, DNA coming from father, half and half, right? Then you are still wondering why they are not same.
Yeah.
The same DNA is coming. Can anyone explain reason for this? There is a reason for that. If you understood very well the previous lessons, you can find the reason why their DNA is not same. Because father and mother DNA is same.
Oh, because of the genetics of early.
No, when they make the gametes produced by meiosis. When they produce gamete by meiosis, it brings the variation to the. Remember that crossing over random assortment, it mixes up all this DNA. It's a random process. So making the gamete cells. So when they make the gamete cells, the gametes are different. I mean, then you can also assume, okay, if a family, mother and father is same, but children are different. They have siblings, they are different. The reason is the story is different, the meiosis is different. So meiosis has a big changes. So then they don't get, but I think on the other hand, if you're trying to find it out, the biological father in case this or for this one, that may be a little bit difficult because of the father and mother, they are same DNA. They have same DNA.
Yeah, so that's seeing those things in something called pattern reports. So like they're checking DNA to see who the cause is.
Yeah.
So if something like this happens, they can't do it.
No, they can't. I think doctors don't recommend marriage between twins, complete twins. It's very difficult. They can marry, but they, as long as they don't care this kind of issues. There is change in expression. Sequence, no change. There's a change in expression. Any transcription factors at RNA polymerase. And pre-mRNA, mRNA, and that's a protein. It's produced in the ribosomes. All right. So next, we want to find it out where we can find these changes, where the changes happen, right? Now, when it try to address that question, it's largely we can put everything into one bucket called environment, surrounding of us. So, what is then environment? Environment is many things: food, lifestyle, behaviors, air, job, the family. We have so much of things when we call environment, but it's very hard to exactly figure it out. A, a very specific situation. Now, last time I showed you a picture that is very specific, right? That picture you can clearly see. Grandmother is smoking, and the chemicals can affect her baby, and also a chemical can affect developing egg cells. So baby and the egg cells. So now we can understand cigarette has so much of chemicals. The chemical can influence the developing baby and her egg cells. And then when she has her daughter and her daughter is pregnant, then this egg cell is influenced. As a result of that, the grandchild also get influenced. So it can influence like this many ways. Then probably who knows that some of the factors that the grandchild got can also influence her egg cell and the next generation influence. So it's a generation to generation. This kind of a changes can pass. So it's not from a single person. It can pass from generation to generation. But in epigenetics, scientists also come up with the idea they can reverse this epigenetic modification, right? It's a it's a little bit of questionable how that can be. Let's say like this one example now, a family from our country is going to USA, and they live in USA. Then the background of the father and mother is very calm and quiet background they had. Like the families are religious, and they are very shy people. Let's say to make it. The father is somehow very good in IT, so he can find a good job. And then still he's very shy guy. Less he's talking less. He's not very social. And now he's stay in USA. Now his son is going to USA school. USA school, the kids are very active, and most of the kids that class is very aggressive, active, and so much of fun or whatever. But this boy still behave like their father. Is it epigenetic, or is it behavioral, or is it what? Because we can argue like the kids can mostly take on the environment what they exposed, not rather than parents, but the school. The school environment is different. The school environment is different, so we can assume that they can get more influence from the school rather than the family. But he still behave like his father. So then we have a question: is it epigenetics? Because still it's not changing from the environment. This is difficult to change. Maybe he's changed. I mean, another example is he changed. So then it changed the epigenetics. Like that, they can say that can be the epigenetics can be changed. Okay, now we still wants to know which level this happened, which level, how this happened. Right now, answer is like this. Answer is scientists found one method. Let's say method number one, they adding some kind of a chemical modification to DNA. Let's say that's a chemical, and this chemical bind to the DNA. So that binds to the DNA molecule, called tags, chemical tags. But how? So let's say this is the environment, and this is the chemical, and I can also name this chemical methyl group. You know, methyl group is CH3, right? This one. So now the methyl group adding to this DNA molecule. Now again, if we want to find the complete story of this, that means how this exactly happening, what is the environmental influence